Yeah, as you heard from Jan, we are working with the cell secretome of peripheral blood mononuclear cells. And it's important actually to know as much as possible about the mode of action or mechanism of action such a secretome can have. So what we are investigating actually are the functional effects of the test substance on the living organism or on cells, on the cellular and on the molecular level. This is important to really understand, and it's also important for the regulators to go further into clinical trials. So this is actually just to remind you what we are working with. So the secretome of blood of mononuclear cells from the blood, which we then lyophilize to get the powder to make it more stable. And as you can imagine, the mode or mechanisms of action from such a secretome is tremendous. There are so many different processes going on, and actually it's not possible never ever to define all these mechanisms, but we are working hard to elucidate at least some or the major mechanisms of this secretome. So as I said, it's quite complicated already, but we are still doing it more complicated, for example, by irradiating the cells. And as Jan said, you can call it secretome or paracrine factors. Actually, we also call it secretome, but maybe it's more accurate to call it paracrine factors because secretion of proteins is an active process. So if you irradiate the cells, you have a lot of cell death, and then you get also passive release of proteins. So actually, it should be paracrine factors, the right name for this secretome. So we call it still secretome. Yeah, as I said, we also do irradiation. Why are we doing this? Because we could show in an initial experiment that the secretome of the living cells has a kind of effect. It was quite nice, but here you're in the heart attack or myocardial infarction in rat animals. But when you irradiate the secretome, then the effect is much stronger. So this suggests that there is a lot of factors which are tissue regenerative produced after irradiation. And when you look at the transcriptomics of these irradiated cells, you see that the gamma irradiation induces many, many genes. So more than 4,000 genes are regulated by irradiation, suggesting that there's really a lot of newly produced factors which can contribute to the regenerative effect of our secretome. So then the other thing is, why is it so complicated to find out all the mode of actions? Because we are not only dealing with secreted proteins, such as cytokines and chemokines. Our secretome contains much more components. For example, lipids. They are known for several biological effects. Also extracellular vesicles, as we have heard previously, like microparticles and exosomes, and also nucleic acids. Just to show you some of the factors which are in our secretome, these are cytokines and chemokines, and don't show you single spots, but you can see here proteome profiler. There are, on this spot, there are 100 proteins, and there are maybe more than 60 or 70 cytokines present in our secretome. As with regard to the lipids, we find different lipid classes. Most importantly, the phospholipids, which are known for a lot of biological functions. And also, as I mentioned, extracellular vesicles like small microparticles and also exosomes. But you will hear a very nice talk in the afternoon by our PhD student, Anja Wagner, and she will present detailed information mechanisms of all these subclasses of our secretome. So another thing which makes the secretome much more complicated is that we are not working with a single cell type. We are working with PBMCs, which consist of many different cell types, such as monocytes, T cells, NK cells, and B cells. So this is another factor which makes everything more complicated. And the last thing which I want to mention, which makes it again more complicated, is that we recently discovered that the gamma irradiation of the PBMC does not only induce apoptosis. There is also a second type of programmed cell death called necroptosis, so the programmed necrosis, which also could have a high impact on the released factors from the PBMCs. And we are doing in the moment studies. That's a study of Dr. Siemader from our lab, where we block apoptosis or we block necroptosis and analyze how the secretome changes and how the effects of the secretome changes. So there's still a lot of work to be done. So as I said, we are working with PBMCs, and this might be very important because what we see in many different experiments is that our secretome has the highest potency when we use all the cells together and not single cell types. For example, here is shown the different kinds of cell death. When you isolate these cells and irradiate them, they behave completely different as the combination of all these cells, suggesting that the secretome composition is also completely different. This is seen here, for example, for some of the cytokines which are expressed in the secretome. Here you can see again the single cell subtypes. And for example, all these proteins which are circled in red, they are only present if you culture all the cell types together. So none of the cell types express these proteins by itself, only when they are cultured together, suggesting that they are influencing each other and which might be very important for the composition of the secretome. So to elucidate a mechanism of action, what you normally have, you have normally a pure substance, and then it might be quite simple. This is, for example, a substance which is widely used in dermatology. It's the so-called all-trans retinoic acid. So to find out what's the mechanism of action, you just have to know it's binding on a specific receptor which leads to the activation of some transcription factors, which induces this and that process in the cells. But of course, for the secretome, it's much more complicated, and Max called it a soup. We would call it an elixir, maybe, with magic functions. Yeah. So I think we could work for our whole life, and we will never find out the whole spectrum of action. But as I said, we do our best, and in the next slides, I will present you some of the mode of actions which we have already elucidated. For example, one of the major effects of the secretome is effects on blood vessels. So Jan mentioned already studies in wound healing. So when we looked into the mouse wound, you can see that there are much more vessels. This is 7 days after wounding, when you treat the mouse wound with APOSEC cream. And here you can see the numbers. So the vessel formation and vessel area is strongly increased in vivo in a mouse experiment. Then we checked this in endothelial cells in vitro, and we could see that the proliferation of endothelial cells is very strongly enhanced. This is even stronger than with VEGF, which was our positive control. So it has a very strong angiogenetic activity, and it can also induce the building of tube formations in matricial assay. So then we did ex vivo experiments. So we have here aortic rings, which are placed on a matrix, and then you can see the sprouting of the endothelial cells. And as you can see here, the secretome has a very strong effect. This is also stronger than the VEGF control. And here we did one experiment with blocking with VEGF, so with anti-VEGF. So you can see that you block a part of the effect, but not all. So there are different angiogenic factors present and important for the function. Another very important effect is related to the vessels is vasodilation. So we could show this also in aortic rings, or actually in coronary artery rings, that the vessel dilates after application of APOSEC. So this is actually a very important process. And how is this going on? So what we also could show is that the treatment of the cells strongly induces the production of iNOS and phosphorylation of eNOS, which then is important for NO production, which could then again induce the vasodilation. And also some other factors are upregulated, which also contributes to this mechanism. So next mechanism, also very important with regard to vessels, is platelet aggregation. So if the vessel is blocked after an injury by aggregated platelets, this is actually not good for tissue regeneration. So we investigated, is there an effect on platelet aggregation? And this is seen here. If you induce platelet aggregation, you get a very high curve. And the second curve is treatment with APOSEC. And here, again, control treatment. So there is a very strong effect of APOSEC on platelet aggregation, suggesting that this is inhibited. This is, of course, in vivo, but suggesting that it's also inhibited in vitro. And we went a step further and analyzed how this could function. And there is a very prominent protein called WASP. And if this is phosphorylated, it can inhibit aggregation of platelets. And this is seen here. So the WASP phosphorylation is induced by treatment of APOSEC. So coming to another mode of action regarding the effect of skin cells, of APOSEC on skin cells and barrier formation, also very important. This is the slide which Jan showed already. When you look at the H&E staining, you can see that the epidermis was regenerated much faster. So after 7 days, you got already more or less a fully covered wound with a new epidermis. And why is this the case? So we investigated again in vitro the effect on skin cells. What we found was that APOSEC induces the migration of fibroblasts and keratinocytes, but does not induce the proliferation of these cells. So this is just a migratory effect. And very importantly, this is also a slide which showed Jan, when we transplant in a wounding model, when we transplant skin and investigated the regeneration of the skin after 10 days, we saw that the epidermis was nicely formed. This was actually not very convincing. After 10 days, it was more or less clear that the wounds are completely healed. But this is even more important, that the staining for a late keratin. So as you can see here, this is the original transplanted skin, and this is the newly regenerated skin in our model. And here are two controls. As you can see, the newly regenerated skin in the control does not express this late keratin, suggesting that the epithelial barrier is still not fully recovered. When you look here in the APOSEC-treated skin, you see that this keratin is very strongly present, suggesting that the epidermal barrier is much faster recovered after treatment, which is, of course, very important for the patient. And then we also checked antimicrobial activity. This is also an issue in wound healing. Wounds are very often, or get very often infected with different strains of bacteria. So we checked if our secretome has properties as antimicrobial treatment. And we checked first for the presence of antimicrobial peptides. Here you can see some of them. They are actually present in very high amounts. And even maybe more important is that when you inject APOSEC into rats, they strongly induce endogenous AMP production. So they have more or less their own protective effects upregulated. And just to show you that this is really effective also on bacteria, we incubated here bacteria with APOSEC, different kinds, gram-negative here in red and gram-positive bacteria in blue. And then we we plated the bacteria on agar plates and counted the colonies which came up. And you can see here very nicely that there is a very strong inhibition of bacterial growth after APOSEC pre-treatment, suggesting that APOSEC directly kills or at least inhibits the growth of different bacterial strains. So, coming to a next very prominent function of APOSEC, and this is the tissue protection. You have seen this slide already many times. In the myocard, you can see that there is much less tissue destruction. Is this actually also a tissue protective effect on cardiomyocytes? So we studied this in vitro in primary cardiomyocytes. And as you have seen already before in the talk, that cardiomyocytes react to hypoxia with cell death. In our case, we used starvation as induction of cell death. And this could be very efficiently inhibited by the addition of APOSEC. And as for the mechanisms, we could show that anti-apoptotic proteins are strongly induced in these cardiomyocytes, and also protective transcription factors, for example, CREB phosphorylation, but also the well-known ACT pathway, for example. They are all strongly upregulated in these cardiomyocytes. But not only in cardiomyocytes, we also could show this, for example, in neurons, that APOSEC also induces the upregulation of, or the phosphorylation of CREB, which is a strongly neuroprotective transcription factor in primary neurons and in glial cells, which suggests that it also acts cytoprotective there. And not only cytoprotective, it also led to the outgrowth of neurons in a primary culture, as you can see here. And as I showed you before with the antimicrobial peptides, it is also the case here for neurotropic factors that APOSEC injected into a rat induces their endogenous production of cytoprotective or neuroprotective factors. And we can see this effect then also in different in vivo models. Here it's a spinal cord injury, which you can see here. When you give APOSEC to these animals, this injury is very strongly reduced. And the same was seen in a stroke model. Here you can see in white the damaged tissue, and this is here strongly reduced also in the stroke model in the brain. So tissue protective also in vivo. Yeah, so coming to the end, actually, due to time reasons, I cannot show you a lot about the immunological effects. I just want to show the picture which also showed Jan before, that we could show in an myocarditis model that the inflammation is massively reduced after treatment with the secretome. And in the moment, we are doing many, many experiments regarding immunological functions. And this would be a talk by itself. So we could show that it acts on T cells, on dendritic cells, on mast cells. So many different effects, which are very interesting, but we are still on the way to fully elucidate these effects. So to summarize, actually, what I've shown you today is that the secretome contains many, many different components and factors, which makes a full elucidation of the mode of actions actually impossible. But we could elucidate at least some of the main effects, which is neoangiogenesis, vasodilatation, antimicrobial activity and reepithelialization in the wound healing models, then platelet inhibition, and the cytoprotective effects and also immunomodulatory effects. And that's the end of my talk. I want to thank everybody who contributed to the work. It's too many people to put them on one slide, so thanks to everybody who contributed and to you for your attention.